비즈니스 널리지를 주제로 콜롬비아대 마틴 첼피 교수님의 강연이 있겠습니다. 큰 박수로 맞이해 주시기 바랍니다.
Thank you very much. It's a great honor to be here, and I hope I get a chance to talk with people afterwards and get the chance to meet more people here. It's been a very nice visit so far. What I was asked to talk about was what were, in a sense, the science education, what should science education be to be able to cope with this fourth industrial revolution, to be able to produce people that will make the new discoveries of the future. And when I was thinking about that, I happened to find an article written in 1939 by this man, Abraham Flexner, in which he says, "I sometimes wonder whether our conception of what is useful may not have become too narrow to be adequate to the roaming and capacious possibilities of the human spirit." He wrote this in 1939. Now, I have to tell you a little bit about Abraham Flexner. He was a remarkable individual. In 1910, he was responsible for a very important report called the Flexner Report, in which he outlined what should happen in the United States for medical education. And for now over a century, we are still following the guidelines that he laid down, he and his committee laid down for teaching medical students medicine. He then, among other things, in 1929, 10 years before this quote, he had the founding idea to set up what is called the Institute for Advanced Study at Princeton. This is where many, many notable mathematicians and physicists, and now other people have studied, and I'll give you a quote where he describes the Institute in a little bit of time. And I was quite taken by this article that he wrote. It was in 1939. Let's see if I can get that. Whoops, went too far. Called "The Usefulness of Useless Knowledge." He wrote this for Harper's Magazine, and he said from a practical view, intellectual life is on the surface a useful form of activity in which men indulge because they procure for themselves greater satisfaction than otherwise obtainable. In this paper, meaning this article that he wrote, I shall concern myself with the question of the extent to which the pursuit of these useless satisfactions proves unexpectedly the source from which undreamed of utility is derived. And I think his words are as applicable now as they were applicable then. And in this article, he talks about going to the George Eastman. Now, George Eastman was the founder and the head of the film company Kodak and camera company, and he was a very wealthy man, and he was talking to Flexner about what to do with his money. And he said, "I want to give my money to where it is going to be most useful, to promote people that are doing useful things." And so Flexner writes that he asked Eastman, he says, "Mr. Eastman, who do you think is the most useful person in terms of science?" And Eastman says very quickly, with no thought at all, he says, "Guglielmo Marconi." Marconi, the inventor of the radio, wireless radio, he gave mankind he showed the greatest usefulness. At which point Flexner says the following. He says, I surprised him by saying, whatever pleasure we derive from the radio, or however wireless and radio may have added to human life, Marconi's share was practically negligible. I shall not forget his astonishment on this occasion. He asked me to explain. I replied, Mr. Eastman, Marconi was inevitable. The real credit for everything that has been done in the field of wireless belongs to Professor Clerk Maxwell and Heinrich Hertz. In other words, the people that discovered the underlying science of electricity. And that's really the theme of this article, that it's not the last person who does it. That person may get fame, but it's the people that actually gave us the understanding that are the real people that push science and understanding forward. And this has been said by many people. I happened to run across a quote by by Mark Twain, who said in a letter to Helen Keller in 1903, it takes 1,000 men to invent a telegraph, or a steam engine, or a phonograph, or a photograph, or a telephone, or any other important thing. And the last man gets the credit, and we forget the others. Well, if this is the case, we have to promote the others. And that's really my, I guess, the topic of this talk. I want to sort of say this a third way. The company Quantsoft, and you can see their URL at the bottom, made this very difficult to read poster. But what this poster is what were all the things that were needed to make the iPhone. And you can look this up online. This is the top. So for an iPhone, you need Wi-Fi, you need a camera, you need a battery, you need an LCD display, you need internet and World Wide Web, and it goes on from there, and computing. And they document all the discoveries that needed to be done for this product to be made. And if you go a little bit deeper into the poster, you see that there's an astonishing number of people. Here's some things that are recent. I'm having a little trouble with the pointer. You can't see it. In the 90s, but this is going back to the 1800s. But this is just the very top of this poster, because if you go even further down the poster near the bottom, you find that there are things that were done almost 3,000 years ago, in which there's discovery of the first lens, there's discovery of geometry that's needed to sort this out, and so on. It's a fascinating collection of all the thousands of people that had to make major discoveries in order for this one product to be made. And so how do you promote these individuals? Now, I want to give another example, and that's the example of what I've been involved in, and that is this wonderful protein from the jellyfish, green fluorescent protein, or GFP. This is actually, for me, a very sad slide, because I heard about 4 hours ago that Osamu Shimomura, who shared the Nobel Prize with me in 2008, died on the 19th of this month. So I'd sort of like to dedicate this talk to him and the amazing things he did as a scientist. I have to tell you a little bit about the history of green fluorescent protein, and a little history about Osamu. Osamu, at the age of 16, you wouldn't say he had a promising career in any way. At the age of 16, he was told he had to quit high school and go work in a paint factory. He left school, he crossed through the valley over to past the mountains that were near the city he grew up in, and he started working in a paint factory. Why did he have to leave school? Well, it turned out that was 1945, and it actually was a bit of good fortune for him because the city he left was Nagasaki, Japan. And by leaving the city, he was saved when the atomic bomb destroyed the city. He went in and rescued people, but he couldn't go to school. There was no school to go to. And finally, he does go to school. He gets an undergraduate degree, and he goes to work as a technician at Nagoya University. Now, he's an astonishingly talented person. And the head of the laboratory there gives him a project that the head of the lab wanted to get an answer for, but what he did not tell Osamu was that several other people had tried to answer that question, and they had all failed. He didn't tell Osamu that, and Osamu was able to get it to work. As a result of this astonishing work, and I should mention that the work was about a problem that has absolutely nothing to do with human biology. The question was, how do certain organisms generate light? In particular, a small crustacean called Cypridina. Now, the thing is that there are many organisms that generate light. There's fireflies, there's glowworms, there's bacteria and fungi and jellyfish and fish and crustaceans. But they do it biochemically very differently from each other. And so to understand how that particular organism generated light, he had to do some very intricate and involved biochemistry. But it worked. And as a result, he is given a very unusual present. He's just given his PhD degree. He also gets invited to come and work in the United States. And he starts out on what became his life's work, which was to understand how, at a biochemical level, all different types of organisms can generate light. And so he started working on this jellyfish. Now, it was a wonderful problem. How does this jellyfish produce light? I can also tell you that most people didn't care. But he cared, and he started studying this. And as he studied it, it's really not a very good story about the scientific method. Because we usually tell students, you know, if you want to do science, you should come up with a hypothesis, a problem, come up with the hypothesis, and test your hypothesis. Well, he wanted to isolate the protein that made this jellyfish produce this beautiful green color, produce light. And he did the preps over and over and over again. And every time he did the experiment, it failed. It failed completely. Nothing worked at all. One night, he had spent the whole day on another failed experiment. He was tired. He wanted to go home for dinner. And he decided to clean up and take the preparations that he had, and he threw them in the sink. The sink also had some seawater, some other things in it, but he was just throwing these things away. He turns off the light, and he's about to leave, when he happens to look at the sink, and he sees that it's glowing brightly. None of his preparations had glowed at all, but this was glowing brightly. He wonders about that and says, maybe there's something in the seawater that I never included in my reactions. And he thinks about that a little bit and says, it must be the calcium. And sure enough, he repeats the experiment, And every time he adds calcium to the prep, eventually getting the protein, a quarant, it generates a flash of light. So calcium was the missing component. Now, the physicist Enrico Fermi has a wonderful, wonderful saying that goes something like this. He said, if you do an experiment and it confirms your hypothesis, then you've made a measurement. But if it does not confirm your hypothesis, then you've made a discovery. And you see, you have to do a lot of experiments to make these discoveries, and you never know where they're going to come. It's only after you make the discovery that you have the hypothesis, and then you do those experiments. But the discoveries come when you least expect them. But fortunately, he was allowed to make that discovery. But he wasn't finished there because, unfortunately, the jellyfish produces green light, and this reaction produces blue light. He tried adding calcium to all the other samples that he had. Nothing produced green light. And he thought about that for a little while, and he just says, well, maybe it doesn't generate the light. Maybe it converts the blue light to green. Now that's fluorescence, if you take one color in and give a different color out. And so he goes and he has a handheld lamp, and he looks at all of his samples, and sure enough, a completely different protein. If you shine blue light into it, it glows green. And when he wrote up his paper in 1962, he wrote a footnote. That footnote said, there is a second protein. I'm going to call it the green protein. And if you shine blue light on it, you get green. He had no idea that there were such things as fluorescent proteins. No one did. Another accidental discovery. Now, we, of course, don't call it the green protein. We call it the green fluorescent protein, or GFP. And sure enough, if you add GFP to a quarant with calcium, you wind up with green light. Another completely accidental discovery. I should also parenthetically point out that as far as I know, this is the only time that a footnote won its author a Nobel Prize. So you don't have to write a lot. It just has to be very meaningful. And this was a very meaningful discovery. But people sort of ignored it. They knew about the protein, or some people did, but it wasn't in the mainstream. People sort of forgot about it. I was fortunate in 1989 to hear someone talk about this. And I'm a geneticist. I work on an animal that is transparent, and I look at the development and function of the nervous system, how nerve cells develop and how they work. And when I saw this, I realized that we could use this wonderful protein and put it inside living organisms and be able to see things happen in the living organisms. In other words, we would be able to see, simply by shining blue light, wherever that protein was made, we would see green, so that it would serve, in a sense, as a lantern, allowing us to see things happen. And I got very excited about this, and with several other people, we did the appropriate experiments to test this idea. And 30 years after Shimomura's discovery, we were able to show that this protein could be used to label, to mark parts of the cell, or cells, or parts of a cell, or different organisms. So I want to tell you a little bit about that. But here's an example of someone who had the freedom to make discoveries Certainly, I think by anyone's criteria, this would be useless knowledge. It's proven to be exceptionally useful. So here's some examples of GFP put into a number of different organisms. This is the tiny, up in the upper left-hand corner, there's the tiny worm that I work on, showing every one of its nerve cells. Below that is fruit fly on the left, and below that is alba. Since this is a forum on science and culture, let me say a little bit about this rabbit, this bunny, alba. This was commissioned from a French biotech company by the Brazilian artist Eduardo Koch. And he wanted to have a green fluorescent rabbit so that he could go to his art shows and get people talking about the connections between art and science and art and technology. In the middle, at the top, we have canola plants. Below that, mice that have had this in it, and below that, zebrafish. And on the right-hand side, there are certain cells, and you can see, for example, in the bottom, this is a Purkinje cell. That's a cell in the cerebellum in the back of your head, and you can see how the green fluorescent protein has gone everywhere within this cell. Now, one of the things I find particularly interesting about GFP, and I think about any major basic discovery, is that they give rise to more basic information, more fundamental ideas. They are used for applications, and they come up every once in a while. They're used for things that you would never imagine, or at least I would never imagine. This truly is the usefulness of useless knowledge. Let me give you some examples. People have been looking in cells for over a century with various staining methods and so on, and we've come to believe that there are parts of the cell called organelles, the mitochondria, the ribosomes, and so on, and then there is the soup that they are swimming in, and that soup is called the cytoplasm. And the cytoplasm was thought to be just a solution, literally a soup. But in 2009, researchers found that it wasn't a soup. It was a combination of different components. And what you could see there briefly is all these white bits, that's labeled with GFP, and it's like oil and water. They don't mix with the rest of the cytoplasm. They are a completely new component of the cell. A fundamental discovery enabled by another fundamental discovery. But like every fundamental discovery, it also leads to applications, or at least some thoughts about possible applications. It is thought that these components that are separated from the other parts of the cytoplasm, their ability to separate, their ability to be different, something no one ever imagined before, may actually underlie, if it goes awry, the events that take place, for example, in Alzheimer's. And people are studying that. Here's an example of something that if you don't know that something exists, you can't look at what its relevance is to a human disease. So if you want to study human disease, you have to get more and more of that basic information. So another example of applied is this example in which the AIDS virus, HIV, was labeled with GFP and infected a cell. Now, when we usually teach undergraduates about viruses, we often describe what happens to viruses that infect bacteria. And what we say about these viruses is that the viruses build up in the cell, and then at a particular time, the cell breaks apart, and the virus goes everywhere, infecting other cells. Well, if that's the case, then that virus is going to be available for an antibody made by a vaccine to attack it and maybe get rid of it and protect people with the virus. But only if the cell explodes, distributing this stuff more generally. So what happens with the HIV virus? Well, let's take a look. There's a cell that's infected. The green of GFP shows how it has been labeled. And there's another cell that you can hardly see, but you can see a particle there. And you can see that this particle has now just been transferred inside another cell. No explosion, no massive distribution, very little chance for an antibody to interact with the virus as it gets passed from one cell to the next. And that's an important way of thinking about disease and how one would then go about controlling it. And then, as I say, there are unexpected things. One of the most unexpected to me, I don't have a picture of, but I'll just tell you, soon after the Nobel Prize, I was asked to give a talk. And at this talk, afterwards, a man came up and he said, well, I want to thank you for your talk. He was the head of the Zeiss microscope company in the United States. And he said, Marty, you don't know this, but in 1994, before your paper came out, the Zeiss microscope company had decided to stop making fluorescent microscopes. Then your paper came out, and everybody needed a fluorescence microscope. So I want to thank you for saving our industry. And I have a little present for you. And of course, I asked for a microscope, but he didn't give it to me. In any case, that was not part of my plan to save a company and their fluorescent microscope division. Another unusual example, which I will show you a picture of, was some work by a man named Bob Berledge. Now, he did a really, at first, strange experiment, but I think a quite interesting one. And what this was an experiment in which he said, I know that there are some control parts of DNA that can turn on the production of any protein if the signal is the small molecule TNT, the explosive. He said, I want to make a bacteria that will make GFP in the presence of TNT. And so he made the construct, made the bacteria, and sure enough, in a dish, without TNT in the dish, no GFP was made. But if there was any of the explosive, a little tiny amount of it, all of a sudden the cells would start making green fluorescent protein, and they would glow green whenever blue light was shown on them. Now you might be saying to yourself, why would anyone want to do that? What would motivate them to do that? Well, what Bob Berledge knew is that landmines, one of the truly worst components, or one of many truly bad components of warfare, which hurt non-combatants sometimes many years after a conflict. Landmines leak TNT, and he wanted to develop a way of detecting where landmines were. And so he tested his bacteria. He had a friend bury 5 landmines in a 3-meter by 5-meter plot of land. Now, they were not connected. They had TNT in them, but they were not connected because they're not stupid scientists. And he sprayed the area with his bacteria and then came back at night with ultraviolet light to see if he could see where the landmines were buried. And hopefully I'll show that picture. There we go. He was able to actually find them. I know of at least 3 other groups that have studied this, have gotten similar results. But this is not quite ready yet. And the reason it's not ready is that sometimes it doesn't work. We call these false negatives. One does not want to have any false negatives. You want to make sure that if something is no, that there really is not a landmine there. And people are working on that right now. To me, it shows people's creativity and inventiveness and thinking about something I would never have come up with, but I'm really glad that he did. And so I think all of these various regions come out of one discovery. We can say this of many things. For example, the laser. There have been at least 3 or 4 different Nobel Prizes in physics. The one just recently announced on the laser tweezers and short pulse lasers was enabled because there was the laser. Before that, the discovery of ultra-cold temperatures required the laser. The discovery of gravitational waves required the laser. So basic research gives rise to other fundamental discoveries. In addition, it's been applied in many, many different ways. For example, CDs, DVDs, the scanners when you go through the grocery store and look at things, all sorts of applications that people have used lasers for that could not have been imagined unless the basic research was begun. There is an award given in the United States called the Golden Goose Award. Before this is only relatively recently, before this, there was a congressman, William Proxmire from Wisconsin, who every year gave his own prize. And what his prize was, it was called the Golden Fleece Award. The fleece is to cheat people. And his award was for people that cheated the government by wasting the government money on very silly things. And he would find the silliest sounding research proposals, and the press loved him, and every year he would give these awards, until one year he made fun of some research that had actually been used to develop a treatment that had helped something like several million people, and he had to apologize. And then the Golden Fleece Award sort of disappeared. Well, about 6 years ago, some other people in Congress said, no, what we want to do is we want to honor science that maybe at first, well, what they said, the award highlights and honors examples of scientific studies or research that may have seemed obscure, sounded funny, or for which the results were totally unforeseen at the outset, but which ultimately led, often serendipitously, to major breakthroughs that have had significant societal impact. So things that sound funny at first, and then turn out to be exceptionally useful. Things that people were told, don't waste your time on that. Do some real work. So here's a partial list of some of those things. The 3 of us, Roger Chen, Osamu Shimomura, and myself, actually won it for green fluorescent protein, which, after all, is a jellyfish protein. Why would anyone be concerned with that? Charles Towns, who was the person who came up with the maser, which was the precursor to the laser, he won on. And that first year, a 3rd group got it. And this was a group that discovered that coral from the sea, if treated appropriately, turned out to be a wonderful substitute for bone for surgery and helped innumerable people. But they did it by first studying coral, not looking for a bone substitute, but eventually getting there. There's been diabetes medicines that have been found by looking at toxins in Gila monsters. One of the ones I really quite like here is the one, it's the 6th one on the list, rat and infant massage. People were studying how mother rats took care of their newborn infants. And because they wanted to be very careful about Well, they weren't even interested in the mothers at all. They were interested in how the baby rats progressed. And they didn't want the mothers to interfere, so they separated the mothers and the babies. Okay, they're separated. What they found is the babies, even though they were well fed, they were treated nicely, they had warm environments, everything was wonderful, they just started to decline. And then they put the babies back with the mothers, and they found that the mothers licked the backs of the babies. They joked that they wanted to get a technician to lick the backs of the babies and see if it was the licking that was important, but no one wanted to do that job. Instead, they just took a little brush and they brushed the babies. And this massage, this brushing of the babies, made the babies thrive. So what the mothers were doing was in some way stimulating the infant rats to be able to develop appropriately. And then they said, I wonder if this sort of massaging can be helpful for premature human babies. And the answer was absolutely. They have cut down the number of days that premature babies have had to stay in the hospital. They have gained weight, they've improved, they've benefited by recognizing something that came from a study that was looking at baby rats. It has turned out to be exceptionally important for human babies as well. And there's a number of other things here, and I won't even go into all the possibilities. But all of this really goes back to this idea of useless knowledge. Going after studies, thinking about what happens to them, and then leading to new things. Because of the topic of this symposium, the Fourth Industrial Revolution, I was able to get a copy of this book by Klaus Schwab, where he presents this idea of the Fourth Industrial Revolution. As far as I can tell, the first revolution was steam, the second one was electricity, the third was computers, and the fourth is sort of big data and networks. And so what did he have to say about education in the book? And he brings up he says, well, maybe we should use academic institutions. And then he says, no, that's not a good idea. And the reason he says it's not a good idea, I completely disagree with, but this is his reason. He says, career incentives and funding conditions in universities today favor incremental, conservative research over bold and innovative programs. Let me just talk about this for a second. In the United States, we have been very fortunate to have a major funding organization, the Howard Hughes Medical Institute. They fund a lot of spectacular scientists. I've always wanted to have funding from them and never had it, but that's a different story. But I once heard the head of the Hughes say, we let people discover things. We don't do things like the National Institutes of Health. That's just incremental. We really want people to branch out, think about things in a bigger scale. I heard this on the television. I started screaming at the television because I said, what I was screaming was, you don't give your money to anyone who has not succeeded in making discoveries using NIH funding. So I'm not sure that this is really true, that it's conservative. People are conservative sometimes, but in fact, I think most of the major discoveries in the United States have been through discoveries at universities. But anyway, he says, well, okay, what's the next thing that might be important here? Well, how about commercial research? And although he doesn't go into it too much, he basically says, no, that's no good either. Commercial enterprises are not going to do it because they have their challenges. I think one of the challenges is that often commercial enterprises want answers. They want products. They want to have things right away. And sometimes these discoveries take much more time. So he finally comes down to the following. He says, to foster both groundbreaking fundamental research and innovative technical adaptations across academia and business alike, governments should allocate more aggressive funding for ambitious research programs. Equally, public-private research collaboration should increasingly be structured toward building knowledge and human capital for the benefit of all. There's nothing wrong with this, except I'm not entirely sure this is going to work. Because I still think that it's for somebody else to talk about funding what are the ambitious research programs? What are the knowledge that people need? I keep going back to the Flexner idea is that we need more useless knowledge. And I'm not sure any of that will ever get funded unless special care is taken to support that. And so that's my plea here. So Flexner, as I said, started the Institute for Advanced Study at Princeton, where Einstein and Gödel and many other people were. And he said it exists as a paradise for scholars who, like poets and musicians, have won the right to do as they please and who have accomplished most when enabled to do so. In other words, give people the freedom to follow their ideas, and you'll be amazed at what things come out of that. And I want to talk about 3 institutions that have used this idea, only one of which I've been associated with myself. So up here in the upper left-hand corner is the Bell Laboratory. Now, the Bell Laboratory is primarily physicists and engineers, and an astonishing group of people have come through there, and they have made an equally astonishing number of discoveries that have changed the way we view the world and, in fact, the universe. This is a list of all the people from the Bell Labs that have won Nobel Prizes, and particularly towards the top of that is the development of the transistor. A little bit further, there's Arno Penzias and Robert Wilson, who discovered the background radiation of the cosmos. Their story is wonderful. They had this wonderful 30-foot microwave receiver that somebody else had used, and they wanted to use it for some observations. And so before they did their experiment, they needed to calibrate the machine. So they stuck it at night looking right up at the Milky Way, and they got a nice reading. Lots of microwaves were coming from the Milky Way. And then they turned it away from the Milky Way. And it didn't go to zero. It was supposed to go to zero. And no matter what they did, they could never get it to go to zero. Why? Because what they discovered in just making sure that the equipment was working right was the background radiation in the cosmos, which is the first indication of the Big Bang, the remnants of the Big Bang. Now, as opposed to mainly physics and physicists at the Bell Labs, the Laboratory of Molecular Biology in Cambridge, England, has been a biochemistry-oriented place, as its name implies, molecular biology. And its list of Nobel Prizes is about as long as the one from the Bell Labs, including 2 for Fred Sanger for protein sequencing and DNA sequencing, Kendrew and Perutz for X-ray crystallography of proteins, Watson and Crick for the structure of DNA, and going down, one thing I want to point out, even though the Bell Labs no longer really exists that much now, most of those people have left, here we have at the bottom of this list Arthur Ashkin, who developed the laser tweezer and won the Nobel Prize this year, and Greg Winter, who also won the Nobel Prize this year for his work on what's called phage display. And then we have the Princeton Institute of Advanced Study, which is primarily an institute for mathematicians. There is no Nobel Prize for mathematicians, but there is something called the Fields Medal. Every 4 years, they give 4 people this top prize in mathematics. It hasn't been going that as long as the Nobel, but the Princeton Institute of Advanced Studies counts 41 of the 56 recipients of the Fields Medal among the people that have gone through that institute, which is an absolutely remarkable accomplishment. So what is that there about these places that allows for these groundbreaking things to be done? Thank you. So I don't know about the Institute of Advanced Study or the Bell Labs, but I can tell you something about the Laboratory of Molecular Biology, and this is where I want to end my talk. I want to talk about what made this such a wonderful place for all these people to make discoveries. I'm not on this list, although I was there. My work with GFP was after I left, but I was inspired by all of these people. So what was important about this, and what I think is really the answer, or at least one of the answers, to what we still need going into this 4th industrial revolution. In fact, whatever will become the 5th industrial revolution needs to have these sort of research institutes. So what were the things that I found particularly useful? I should back up just a second and tell you what happened to me. I came there as a postdoctoral fellow. Normally, at least in the United States, if you go as a postdoctoral fellow, you work for somebody, they are your boss, you talk to them often. Not here. I went, I talked to the person who was signing the checks, Sidney Brenner, who won his Nobel Prize many years later, 2002. And Sidney asked me, he says, "What do you want to work on?" And I said, "Well, you know, I wrote a proposal about this, but I'm actually now getting interested in this other area. I'd like to work on that." And he said, "That sounds okay." That was the last conversation I had with Sidney Brenner for an entire year. In fact, I was there for 5 years, and I talked to him only 5 times, as did a friend of mine who was in the same situation. And when we were comparing notes many years later, we both said, we talked to him only 5 times in 5 years, and then simultaneously we said, and the longest conversation we had with him was when he had his motorcycle accident and was in a hospital bed so he couldn't walk away from me. In any case, we were given complete freedom. But along with that freedom, there were astonishing colleagues. It was an environment of just great people to talk to. And I learned immense amounts of information and got encouragement and help from just a wide array of really, truly wonderful colleagues. So having a critical mass of people that can interact, to me, is very important. The second thing, the facilities. Anything we wanted was there. If we needed a special microscope, we would be able to get the microscope, we could arrange things. If it was something that wasn't manufactured, they had wonderful shops, they made it for us. So we could get any piece of equipment we wanted. We also had wonderful supplies, anything we wanted. I once went down to the stockroom because I needed to do a chemical reaction that required an ultraviolet lamp. And I went to the stockroom head and I said, "I need a ultraviolet lamp." And he looked at me completely disgusted and said, "What wavelength lamp do you need?" Not, "Oh, we don't have one." It's, "Which of the many types of lamps do you want?" Now, if you have the best colleagues in the world and you have any facility that you want, and all the equipment, and you have all the supplies you could care for, this leads to a problem. You suddenly realize you have absolutely no excuse. If you don't succeed, you cannot blame it on the equipment or your colleagues or the supplies. It is up to you. Some people flourished in that environment. Other people found it too scary. I was very fortunate. I had a lot of help from people. I found it a wonderful thing to do. So it's this support, the overall support of being able to have people that you can interact with, but the complete freedom to work on whatever problem that you want. That is something that I think characterizes all three of these institutes. So if I'm going to conclude anything about what I've said so far, I would say that what we need is to have opportunities for people to be able to follow their intuition. We've trained them enough. They know about these things. But being able to have the freedom to make these unexpected discoveries is, I believe, going to be of continuing importance in the years ahead. Thank you very much.
네, 마틴 첼피 교수님의 강의가 있었습니다. 뭔가 생각지도 못했던 분야 간의 연결과 영향에 대해서 생각해 볼 수 있는 그런 시간이 아니었나 싶은데요. 저희가 강연을 들으면서 질문을 3개 정도 받아봤습니다. 이 오늘 강연을 다시 한번 정리하는 차원으로 생각해도 좋을 것 같습니다. 준비되셨습니까? 네, 첫 번째 질문입니다. 이 학생들이 어떻게 공부하면 노벨상을 받을 수 있을지, 그리고 선생님들은 도대체 어떻게 학생들을 좀 가르쳐 보면 노벨상을 받는 학생들로 키울 수 있을지 물어봐 주셨습니다.
So you're not going to like this answer. I consider the Nobel Prize to be a rather unusual prize. It is not to the best scientist. It does not go to the smartest scientist or the scientist who has the most funding or the biggest lab. It goes to the scientist who has discovered something that has changed the way we think about science or do science. It's my feeling that most of those discoveries are accidents. There are some people that go directly for things, but a surprising number of these are accidents. I told you about Penzias and Wilson, who were just calibrating their machine and by accident found that. I told you about Shimamura, who threw his material in the sink where there was seawater and was able to discover both the bioluminescent protein and GFP. I think his first name is John O'Keefe. A couple of years ago won the Nobel Prize for the identification of cells that help us identify locations in our brain. And his Nobel Prize winning speech starts off, I was doing experiments looking at the thalamus when by mistake I stuck the electrode in the hypothalamus. Of course, that then led to his discovery that gave him the Nobel Prize. So I'm not sure how one goes about getting a Nobel Prize. If I knew it, I would not be here. I'd be back in the lab getting another one because the party is very nice. But these accidental discoveries do come. So I think part of it is give people a chance to work out their ideas, to do as many experiments as they can because they will make those accidental discoveries. And those are going to be very important. I don't think there's anything, I feel for myself that the most important thing that I learned in college was not in my science courses. The most important thing I learned came from my courses in humanities and in social science. And it was the final exams that I thought trained me the best to be a scientist, because those exams were of the following type. We would read a lot of books, a lot of literature, a lot of material for the course, and you were expected to know that. You were expected to know the lectures. But these instructors were very mean. They always asked questions that had never been asked before. And so we had on the final, we had to take all that knowledge we had and mix it and match it and put it together in a way that before then we had not thought about in order to answer that question. I feel that was wonderful training to be a scientist, to be able to make connections where before those connections were not made. So I'm not sure exactly what the training is. I know that for myself, that was an important aspect of my training, to be able to just play with information, to put it together in ways that were unusual. And I still do this now. When I read a paper, when I hear a talk, when I'm speaking to people, I'm always thinking, how could this apply to what I'm doing? Where is the connection between this and what I'm interested in the lab? I think that we do best by helping people find their own curiosity.
네, 이 뭔가의 놀라운 발견을 위해서는 일단은 절대적인 양적인 노력이 필요하지 않나 생각이 들었습니다. 박수 부탁드립니다. 네, 두 번째 질문 이어가 보겠습니다. 기초과학으로 공을 들인 사람이 더 중요할 것인지, 나중에 최종적인 발명을 한 사람이 중요할 것인지, 뭔가 우선순위를 둘 수 있다면 어떻게 둘 수 있을까요? 라고 질문 주셨습니다.
I think it's neither or both of them. We need both. You know, in the United States over the last few years, there's been a big push for what's called translational medicine. Let's take what we know and apply it to human conditions, diseases, and stuff. And as I get older, I like that. I think that's a wonderful idea. But I know, many people have said that, I'm not the first, that if you want to translate something, you have to have something to translate. So you need both the person making the discoveries as well as the person who wants to spend most of his or her time applying those discoveries. It's a little bit like I showed in the slides earlier of all the people that were needed to make the iPhone, or all the people that, the thousands that Mark Twain talked about, but you only remember the last one. What we remember and what's fame, I don't think is as important as all the people that build the knowledge and the cooperation that it takes to build that knowledge to be able to get an outcome. So I believe the error is not one or the other. The error is in thinking that you can only have one and not both, because I think you have to continue to develop the pipeline of ideas and information so that people can apply them. And I tried to give that an example here of the discovery of the new part of the cytoplasm that no one knew existed, and now is changing the way people are starting to think about certain diseases.
네, 박수 부탁드립니다. 네, 다음 질문 한번 다시 이어가 보겠습니다. 녹색 형광 단백질 GFP, 이게 어떻게 실용적으로 쓰일 수 있는지 뭔가 병균 발견 등 이런 것들 설명해 주셨는데, 조금 더 구체적인 실용적인 예시를 더 알고 싶다고 하십니다.
So I can give several examples, but then we would be here for several hours, so I'll give maybe only one. I will tell you how we use it in my laboratory. If you look at the animal that we study, what you will see is nothing. We're interested in 6 nerve cells but all the cells basically look alike. But when we put GFP into the animal, only those 6 cells are green. And so in this otherwise transparent animal, we can see those particular nerve cells. Not only in that animal, but in its progeny, its babies, and in their babies, and in their babies, it gets passed along. We can see the cell. And once you can see anything, you can start to study it. So these cells are very simple. They are a simple nerve cell, they have a cell body, and from the cell body, a long process goes towards the front of the animal. We've looked for mutants that don't have the cells. We've looked for mutants in which the cells are in the wrong place. We've looked for mutants that have more than just that one process. We've looked for things where the process doesn't go forward, it goes backwards. All of these help us understand basic ideas of the biology of how nerve cells grow, and hopefully give us some better insights into the ways that nervous systems develop and connect up with one another. This basic idea is, if you can see it, you can study it, has been the basis of almost countless experiments. It has been estimated that since we published our paper 24 years ago, there have probably been 200,000 papers that have used fluorescent proteins in a variety of ways. There are some journals that you cannot open and not see every single article in the journal. Maybe one will not be using a fluorescent protein, but it's being used to study all sorts of phenomena, not just the ones I gave you an example of. But being able to see things allow that to work. There are certain, we know the energy component of the cell, the mitochondrion. Every textbook that draws a mitochondrion draws it as if it's a bean. That's the picture we have. But when people actually look in the living cell, mitochondria are coming together, they're coming apart. And this fission and fusion that takes place, this coming apart, coming back together, is absolutely important for their activity. You can watch that with GFP. Not only can you watch it, you can say what happens during disease states. Are there things that disrupt that joining and coming apart? And the answer is yes. Without being able to see that in a living cell, we really wouldn't be able to study this process. So, and I could go on for many other examples, but those are a couple.
네. 감사합니다. 이 다음 질문은 아마 오늘 이 포럼의 이 취지에도 가장 맞는 질문이 아닐까 생각을 해보는데요. 이 한국에서는 자연대에 가면 순수과학을 하면 이 취직도 안 되고 돈도 잘 못 버니까 공대에 가라고 합니다. 이러한 현실의 한국에서 우리나라 학생들, 저 한국의 학생들에게 하고 싶은 말씀이 있으신지, 그리고 걱정 없이 자연과학 분야에 종사하려면 사회에서는 어떤 점이 개선되어야 할지 조언을 구하셨습니다.
So I would not presume to tell students what they should or should not do, but I think that, as I said before, we need both. We need people that are going to make the basic discoveries, and who will often think about what the applications will be, and we need people that will be more concerned about the end step of applying knowledge. That what we need is a lot more communication among these people. Now, I've been a university professor my adult life. Actually, before I was a university professor in graduate school, I was a high school teacher. But so I'm maybe not the person to talk to about making money, but I want to talk about, in the United States, we have something similar, and that is, but not engineer versus basic research. What we have is it's fairly common to hear somebody say, They'll find out that their son or daughter is studying history. And they'll say, "Oh, why are you studying history? You can't make any money." Well, that's okay. But that's the image. Oh, if you study something like this, you cannot make money. You cannot, because people somehow think that succeeding is making money. Well, in the 1980s, I met a really remarkable young woman who had been a history major, and then she took a master's degree in oral history to be able to interview people and talk to them about themselves. And then she started her own company. And her company was a very interesting company because in the late 1980s, this was when all of the dot-coms were starting, all these new companies were starting. And these people that started their companies were very proud of themselves. And she went to them and says, "I'm a historian. I can write the history of your company. I'll interview all the people and get the pictures, and I'll produce a book that will be the history of your company." They loved it. And that's what her business was. Her business was using history. So to me, it's not really a question of, is this a bad thing to study? Is this a better thing to study? It really is how people apply whatever they learn and do. So, but I don't know what to tell people. I'm grateful that I never was told what I should do or shouldn't do as I was growing up. So I'm pretty happy, but I'm not guaranteeing this for other people.
네, 미국의 부모님들도 약간의 같은 고민을 하고 있다는 위안이 된다는 답변이었던 것 같습니다. 네, 마지막으로 질문 하나 더 해보겠습니다. 4차 산업혁명을 대하는 과학자가 가져야 할 덕목 중 가장 중요한 것은 무엇일까요? 하고 물어봐 주셨습니다.
I'm not sure, and I think my talk exemplifies it, that there's anything special that we need. I can tell you that everything I learned when I was in graduate school, all the techniques, all the information, none of that's important now. So there is nothing that I could say, "Oh, you have to study this or you have to study that." I think that what many people have said, one needs a feeling of flexibility, an interest in what's going on, a willingness to make connections that are quite far apart from each other, and also to not listen to too many people of what you should or should not do, including me. I think I'll let it go with that. Thank you very much.
네, 감사합니다. 네, 질문은 여기까지 받아보겠습니다. 마틴 첼피 교수님이었습니다. 다시 한번 큰 박수 부탁드립니다. 네. 지금 카카오톡과 함께 이제 객석에도 질문이 많을 텐데요. 오늘 시간 관계상 이 모든 질문들을 받지 못한 점 양해의 말씀 부탁드리겠습니다. 네, 벌써 점심시간입니다. 점심시간 이후에도 1시 반부터 글로벌 강연이 이어질 예정입니다. 식사 맛있게 하시고요. 늦지 않게 오시면 되겠습니다. 오후 강연에는 4차 산업혁명 시대에 맞는 다양한 강연들이 준비되어 있는데요. 점심 식사 이후에 1시 반에 다시 뵙도록 하겠습니다. 감사합니다.